In this video, we're going to talk about how new species are formed. Now, when defining different species, which organism belongs to what species, scientists use different criteria. So there's really not one unified form of defining species that works for every situation. There are various different kinds. So let's take a look at what they are. The first part is morphological. It's the morphological species concept, meaning that organisms are placed in the same group of species if they show similar physical features. Now, this has its limitations because sometimes there are organisms that have similar features, but they really should not be placed in the same group of species because they really can't reproduce and produce fertile offspring. Then there's the ecological species concept, which scientists use to place organisms into the same group depending on whether or not the niche environment to which they have adapted are similar. The phylogenetic species concept refers to building a phylogenetic tree based on the fact that whether a group of organisms share a common ancestor. So organisms are divided into the same group based on ability to create the smallest set of organisms that share an ancestor and can be distinguished from other such sets. Finally, it is the biological species concept, which divides organisms into species based on ability to mate and produce viable offspring. So if there are organisms that can mate and produce offspring which are fertile and viable, who can go on and for the next few generations produce fertile and viable offspring, then those species are put together. In this series of slides that I'm going to show you and the concepts that I'm going to talk about with respect to mechanisms of formation of new species, we're going to concentrate on the biological species concept. Now, biological species concept, it has applications to many situations, very widespread, but it also has limitations because you really can't apply them to fossils, for example. Or organisms that reproduce via asexual mechanisms. So in that case, if you can't use this, then you have to go to the other characteristics. Specifically, this phylogenetic species concept is really useful. But for this presentation, we're going to focus on mechanisms that give rise to species based on the ability of being able to reproduce and produce viable and fertile offspring. So there are different types of speciation. The first mechanism I'm going to talk about is called allopatric speciation, meaning that formation of new species because organisms were separated by geography. So allo means different, patric means fatherland. Organisms that used to live in the same area, geographical area, but for whatever reasons they're separated, and because they are separated geographically, they accumulate changes, adaptations to their environment, and then over time they become so different that they will no longer mate and produce fertile offspring with each other. And a great example of that was with the different species of finches that Darwin observed on different islands, on different Galapagos islands, even though they are descended from the same common species from the mainland, but when they distributed to different islands because they were exposed to different environments, or after a while, they diverged into different species. And this was clearly shown by this modern experiment by Peter and Rosemary Grant. I will post a link to the video after my video and also in the description of the video. Basically, Peter and Rosemary Grant were able to show mechanisms, things that lead to formation of different species between birds that lived in different islands. So, watch the video shown in the previous slide and describe observations by Peter and Rosemary Grant and their conclusions about what factors led to speciation of finches. Now, unlike allopatric speciation, sympatric speciation refers to formation of new species even though organisms live in the same area. So you don't geographically separate the organisms. So if you don't geographically separate them and you don't prevent them from mating, what causes organisms to diverge, to become separate species? So that's sympatric speciation. And one mechanism is autopolyploidy, meaning that an organism just automatically increases the sets of chromosomes that it has. And this actually happens in plants quite a lot. So autopolyploidy results from when mitosis is not followed by cytokinesis. So the organism doubles its genetic material, but then you keep the chromosomes together. So then you have a parent which is diploid, but an offspring becomes a whole new species because it has twice as many DNA material, and it just becomes a totally different organism compared to the parent. Another form of species formation is alloploidy in the sympatric way, and that happens a lot actually in plants when, first of all, errors happen during meiosis, so sister chromatids or homologous chromosomes don't separate, so an individual species produces a polyploid gamete. So you have more chromosomes than would normally be present in a gamete. So that particular gamete, it happens to kind of come together and fuse with a normal gamete from another plant, and then you produce a new organism, and that new organism produces a polyploid gamete. And then polyploid gamete again in turn is going to fuse with another normal gamete, and you get another totally new organism that has way too many chromosomes present. So you can see that the new species is totally different from what you started with species 1 and 2. So polyploidy and non-disjunction, this is something that happens in plants all the time, and that's one of the mechanisms by which plants evolve. Another example of sympatric speciation are these fish that have been observed in a lake in Nicaragua, and the genetic evidence shows that these two types of fish, they basically arose from the same fish, but then over time they ended up diverging because the different groups of fish ended up living in different parts of the lake, and because they lived in different parts of the lake, then they adapted to their new environment, and they developed new characteristics, which you can see in their new types of lips. And then this is considered to be an early stage of speciation. Okay, so other types of mechanisms that causes speciation are barriers that happen to mating and producing vital and fertile offspring. Based on our definition of biological species concept, if you prevent formation, if you prevent mating and formation of viable and fertile offspring, then that leads to keeping those species separate. So imagine you have frogs, and normally the same species of frog, they mate and then they produce baby frogs, and then baby frogs grow up and produce, they go on they become adults and produce their own offspring. Now, at each of these, there are multiple different steps. These barriers to mating can happen. It could be happening before even these organisms can mate. So there could be pre-zygotic barriers. So even if there are barriers which would prevent these frogs from mating, then if they can't mate over time, they're going to diverge and their gene pool separates. Or you can have post-zygotic barriers. For whatever reason, either the offspring does not survive, or the offspring survive but cannot produce their own viable offspring. So what are some examples of that? One example of pre-zygotic barrier is temporal isolation. So if there are two different frogs, but they tend to mate at different times. Then we'll also have a gametic isolation, meaning that there's just no way that gamete from one organism can meet the gamete for another one. And the choice of pollinator for plants is a good example. So if a hummingbird only feeds on these type of longer flowers, then when the hummingbird comes in and takes some of the pollen from these plants and goes to another plant, unless the second plant that this bird lands on is also the same type of flower, then the pollination will not occur. So the type of pollinator that a plant uses limits the type of different plants that these different types of plants can mate with. So their gametes will not meet because of the pollinators that they have. Another type of pre-zygotic barrier is habitat isolation. If you have different, let's say, types of grasshopper that they just evolved to live in different habitats, and because their habitats are so separated, they just will never meet to mate. Behavioral isolation is another pre-zygotic barrier. Now, if let's say you have different types of birds, and if the female, for example, in this case, the male has to have a certain dance, and if certain males don't have the proper dance, then the female won't mate with them. So behavioral isolation could be a barrier to mating. Another type of pre-zygotic barrier is mechanical isolation, meaning that the sexual organs of the different sexes have to be compatible. So in this case, the shape of male reproductive organ varies among male damselfly species. So different damselfly species, they have different shapes of their male and female organs, and the only way they can mate, these flies can mate, is that there's compatibility between their sexual organs. There are also post-zygotic barriers. One is hybrid viability. Two organisms may be able to mate, but then their fetus does not survive. An example is mating between goat and sheep. They can mate, but they cannot produce viable offspring. Then we have hybrid sterility. That's when two organisms can mate, they can produce a viable offspring, but that offspring cannot reproduce. And that's an example of mule. A horse and a donkey can mate. They can produce a very viable offspring, but mule is sterile. It cannot pass its gene to the next generation. Finally, hybrid breakdown, that's when two organisms can produce viable hybrids, viable offspring, and then those two hybrids can they're also able to produce the next generation. But then the F2 generation, the generation in the second degree, is not viable. And this happens in some species of rice. So two different species of rice, they mate, they produce hybrids, but those hybrids, they can also mate, produce their own offspring, but their offspring don't survive. Okay, now another question that scientists ask is how fast speciation happens. And throughout all the studies that they have done, all their observations, they have come up and have identified two different models for speciation, which we call, in terms of how fast it happens, how rapidly speciation happens. One is gradual speciation, the other one is punctuated equilibrium, and they're different in the rate of speciation. With gradual speciation, small changes, intermediate changes happen first until new species arise. So in the top example, you do not get the new species that are totally different after a long period of time. First, you form the new intermediate species, then intermediate forms, then the new species arise. So by the time you're here, you have new species, but not in here. With the punctuated equilibrium, formation of new species is pretty rapidly. So rapidly from a founder species, you get two whole new different species that are totally different and separate, and over time, they accumulate maybe smaller changes. So that's the difference. Now here are some true or false questions. These are situations and misconceptions that is common about evolution. First of all, evolution is a theory about origin of life. No evolution basically describes why it is that there's so much unity within diversity. How is it that there is so many different species, and why is it that we have so much in common, and how did we start from one place in terms of limited number of species, and how did we get so diverse? Evolution results in progress. Organisms are always getting better through evolution. No, that's not correct. First of all, with evolution by natural selection, really selecting the different type of organisms depends on the environment. If the environment changes, the selection becomes something different. So just because an organism is perfect for one particular environment doesn't mean it's suitable for another environment if the environment changes. And also we've seen other evolution mechanisms like genetic drift and gene flow, and we know that those are just, specifically genetic drift is by chance and gene flow is by movement between organisms. So that has nothing to do with merit or whether you are not adapted. Individual organisms can evolve during a single lifespan. No, we define evolution in terms of what happens to a population. Remember the percentage of different types of variations that we have within the same population. Individuals don't evolve during a single lifespan. That's just not how we define it. Darwin was the first scientist to suggest that organisms evolve by changing over time. That's also false, because the first person who suggested that was John Baptist Lamarck. What John Baptist Lamarck didn't get correctly was the mechanism. It was Darwin who proposed the correct mechanism for evolution.